Hello everyone, welcome to another video of Knowledge of Life. Today in this video we will give an introduction on enzyme. In this video we will know what is an enzyme and what are they made up of, and we will classify the types of enzyme on the basis of their reaction, and we will also know how the nomenclature or naming of an enzyme is done. So now let's start. So what is an enzyme? Enzymes are thermolabile organic catalysts that are protein in nature and are synthesized by the living cells, but they are active both inside and outside the cell and even in a cell-free extract. So, enzymes acts as biological catalysts that speed up biochemical reactions and are involved in most of the chemical reactions that take place in organisms. There are about 40,000 such reactions known to be catalyzed by an enzyme, but the number may be even higher. And enzymes allow reactions to occur at the rate necessary for life. So, in animal, an important function of an enzyme is to help digest food. Now, digestive enzymes speed up reactions that break down large molecules of carbohydrate, proteins and fats into smaller molecules which the body can use. So without digestive enzymes, animals would not be able to break down food molecules as quickly enough to produce the energy and nutrients they need to survive. So enzymes act as a very important biological catalyst in our daily life. Now, what is a catalyst? A catalyst is a substance which increases the rate of a chemical reactions, but they do not alter the chemical equilibrium. They only shortens the time until it is achieved. So, molecules of enzyme leave reactions unchanged and are not consumed. And like other catalysts, enzymes are not reactants in the reactions they control. They help the reactants to interact but are not used in the reactions. Instead, they may be used over and over again. And unlike other catalysts, enzymes are usually highly specific for particular chemical reactions, and they generally catalyzed only one or a few type of reactions. So now let's look at the picture. As we can see in the figure that there is a red line graph and there is a blue line graph. The red line graph is for the reaction with no enzyme, and the blue line graph is for the same reaction with the presence of an enzyme. And as we can see that when there is an absence of enzyme, the reactants needs a much more activation energy to conclude the reactions, that is to form the product. Now with the presence of an enzyme, there is a need of much more less activation energy than it requires in the absence of an enzyme. So enzymes lower the activation energy necessary to transform a reactant into a product, and the enzyme catalyzed reactions bind with the reactant and facilitates its transformation into product. Consequently, an enzyme catalyzed reaction pathway has a smaller energy barrier, that is an activation energy, to overcome before the reaction can proceed. So, enzymes lower the activation energy necessary to transform a reactant into a product, and in the enzyme catalyzed reaction, an enzyme will bind to a reactant and facilitate its transformation into a product, and consequently an enzyme catalyzed reaction pathway has a smaller energy barrier, that is activation energy, to overcome before the reaction can proceed. So now in this picture we can see the natural half-life of some biological reactions in neutral solution at 25 degrees Celsius. So we can see that without the presence of an enzyme, the biological reaction can take place much more time or a huge amount of time to get completed, such as the CO2 hydration can take place for about 5 seconds, the peptide cis trans isomerization will need 23 seconds, the triose isomerization will need 2 days to get completed, the carboxylic esterase hydrolysis will need about 4 years. the peptide hydrolysis in the C terminal of a dipeptide will need 450 years to get completed, and at the last, that is glycine decarboxylation, will need about 1.1 billion years, which is almost about the age of an earth. So without the presence of an enzyme, the biological reaction would be taking much more time, and it and in our daily life this kind of reactions without the absence of an enzyme would not be possible to get completed. So enzymes are present in our system to maintain the equilibrium of our life, and an enzyme can enhance the rate of the reaction to about 10 to the power 5 times to 10 to the power 17 times, such as cyclophilin can increase the rate of the reaction by about 10 to the power 5 times. Carbonic anhydrase can increase the rate of the reaction by 10 to the power 7 times. Carboxypeptidase A can increase the rate by 10 to the power 11 times. Urease can increase the rate by about 10 to the power 14 times. So we can say that in the absence of this kind of enzyme, the reaction rate will also decrease, and the decrease can range from about 10 to the power 5 times to 10 to the power 17 times. So enzymes are very much responsible to maintain the rate of the reaction and to complete the reaction in a much faster rate. Now let's look at the classification of the types of enzyme present. Now the classification can be done on the basis of two types, that is on the basis of constituents or what are they made up of, and on the basis of the reactions they catalyzed. Now at first let's look at the classification on the basis of constituents. Now on the basis of constituents, an enzyme is made up of. An enzyme can be of two types, that is a simple type and a complex type. Now in a simple type, there is only presence of protein, that is they are only made up of protein, and there are no other chemical groups for their activities other than the amino acid residues of the proteins. So there can be various type of simple enzymes present, such as pepsin, trypsin, ribonuclease, and so on. And in a complex type, there is presence of another additional chemical component required to complete the enzymatic action, which is known as the cofactor, and the protein part of the enzyme is known as the apoprotein or apoenzyme. So a complex enzyme is made up of a apoenzyme and a cofactor. Now the cofactor can be of two types, that is there can be presence of a metal ion or organic molecules. Now the metal ions serve as a cofactor for the enzymes to catalyze the reactions, such as cytochrome oxidase need a Cu2 plus ion, the catalase can need a Fe2 plus ion, the urease can need a nickel ion, or a glutathione peroxidase will need a Ac ion to complete the enzymatic reactions, that is the metal ions serve as the cofactor for the enzymes. Now the another type of cofactor, that is the organic molecule, can be of two types, that is the coenzyme and second one is the prosthetic group. Now the coenzymes serve as a transient carriers or specific atoms or functional groups, and they took part in the enzymatic reactions, such as biocytin took part in transportation of CO2, coenzyme A helps in transportation of acyl groups, flavin adenine dinucleotides help in transportation of electrons, pyridoxal phosphate helps in transportation of the amino groups, tetrahydrofluorate helps in transportation of one carbon groups, and the thiamine pyrophosphate helps in transportation of the aldehyde groups. Now there is presence of another form of enzyme or the inactive form of enzymes, which are known as Zymogens or Proenzymes. They are present in inactive form to protect the cells from autodigestion, which can be caused by the active form of the enzymes, and they are only activated when there is the requirement, such as trypsinogen is converted to trypsin by the interaction of enteropeptidase, and the trypsin took part in conversion of chymotrypsin and other enzymes by the chymotrypsinogen and other proenzymes. So Zymogens are inactive form of enzymes which are get converted when they are required. Now there are many same enzymes which catalyze the same reactions. Those enzymes are known as isoenzymes, but they differ from each other in their physiochemical properties. That is, they have got a different substrate affinity, they have got a different Km or sensitivity to inhibitors, and they are present within the tissues. So, coming to the conclusion of the classification of enzyme on the basis of constituents, enzyme can be of two types, that is a simple one and a complex one, where the simple one is made up of only proteins and the complex one consists of two parts, that is a protein part and a non-protein part. The protein part is known as the Apoenzyme and the non-protein part is known as the cofactor, whereas the cofactor can be of two types, that is a metal ion or a organic molecule, and the organic molecules which are certainly derivatives of vitamins can be of two types, that is a coenzyme which binds freely with the apoenzyme or the protein part of the enzyme, and the prosthetic groups which binds tightly with the apoenzyme. Now coming to another type of classification, that is based on the reactions they catalyzed, enzymes can be of six types, that is oxidoreductase, transferases, hydrolases, lyases, isomerases and ligases, and each of them serves in different kinds of enzymatic reactions. The oxidoreductase does the transfer of electrons, that is hydride ions or H plus atoms. The transferases does the group transfer reactions. The hydrolases does the hydrolysis reactions, that is transfer of functional groups to water. The lyases took part in addition of groups to double bond or formation of double bond by removal of groups. The isomerases took part in transfer of groups within molecules to yield isomeric forms, and the ligases took part in carbon-carbon, carbon-oxygen, carbon-nitrogen bonds by condensation reaction coupled to ATP cleavage. Now let's look how the nomenclature or the naming of the enzymes are being given. There are various type of nomenclature process. One of them is the recommended nomenclature. The recommended nomenclature is simpler than systematic nomenclature but yet being systemic in a way, and the recommended nomenclature is often used in a routine practice, and the name is made by combination of two, that is one substrate and S, that is AAC. One such example is amylase, which does the catalyzing of the hydrolysis of amylase, and it can be the type of reactions and S. One example is dehydrogenase. Now another form of nomenclature is the tribal nomenclature. The formerly used term ferments, that was based on the fact that enzymes are involved in the process of fermentation, is no longer used. First discovered enzymes were given names according to their source of method of discovery, and the names are therefore without any relation to the reaction mechanism, and in this case many end with IN ending, such as the pepsin or thiolin. Now coming to the end, that is the systematic nomenclature. The systematic nomenclature has been introduced by the International Union of Biochemistry, and in this type of nomenclature every enzyme has its own EC number, that is the enzyme commission number, and the EC number is made up of 4 digits which are separated by commas, and the first digits denote one of the 6 main enzyme classes based on the type of the reactions they catalyze, and the second number indicates the subgroup, and the third number indicates the subgroup, and the last indicates the precise position of the enzyme within the subgroup and thus completely characterizes the enzyme. Now let's look at an example to make the systematic nomenclature easy. Let's take lactate dehydrogenase, which has got a EC number or enzyme commission number of 1.1.1.27. Here the first digit indicates the class of the enzyme, the second digit indicates the subclass of the enzyme, the third digit indicates the subclass of the enzyme, and the fourth one indicates the precise position of the enzyme, and it completely characterizes the enzyme. So in the next video we will discuss about how does the enzyme works and other details regarding enzymes. So hope this video will help you, and if you have any kind of query regarding the video, please put a question in the comment box. Thank you.